Hi, hello. Welcome to Physionic. My name is Nicholas Verhoeven. If you're not familiar with who I am, I'm a doctoral candidate in molecular medicine, and I also do research in a cell biology lab. And today we're going to be discussing a particular topic that is going to be quite molecular focused, cellular focused, and it's essentially going to be covering the topic of how does metabolism affect mitochondrial behavior. Mitochondrial behavior is going to be defined by the actions that mitochondria kind of enact. So if they split apart, if they combine together, where do they move within our cells, and how that's dictated by our metabolism. I have a background in metabolism, as most of my master's degree was done at a university that's well known for their metabolism research. But as we're going to go through this presentation, just keep in mind that this is not something that you can really jump to a conclusion section and then just get all the information. I'm going to be getting into the nitty-gritty of things as usual, and this is all going to be based off of not just my knowledge throughout my doctorate and before in my master's program, but also based off of a review. So without further ado, I'm going to have timestamps so that you can skip to different sections if you're interested in particular behavior of your mitochondria, and I'll certainly be covering more topics related to this as well in the future. So the question, like I mentioned, that I'm going to be examining, or that we're going to be examining together, is how does metabolism affect mitochondrial behavior? And the review that I mentioned is called A Metabolic Regulation of Mitochondrial Dynamics. And this actually comes from a JCB review, which is known as the Journal of Cell Biology. It's a pretty reputable, or highly reputable journal that these two individuals, these two researchers, compiled a decent amount of research. This only goes up to about 2016, but still pretty current, although I'm sure there's been some that's been added to this as well since then. And this is not all encompassing of all metabolic regulation of mitochondrial dynamics, meaning behavior. So just keep that in mind, but it's still going to give us some pretty interesting information on exactly what happens within our cells. Okay, so with that, let me go ahead and jump into this. So as usual, I like to have a bit of a background on mitochondria, really just the subject that we're going to be discussing. And in this situation, we're going to be talking about mitochondria. So let me actually, I put my video up for a second, and now I have to remove it so you can actually read the rest of it. So mitochondria have many purposes beyond just energy production. People think of ATP generation when they think of mitochondria, the powerhouse of the cell, right? If you've been following Physionic for a while, you know that story. But there are other facets of mitochondria that are incredibly important, at least as important as the energy production value of the mitochondrion. So energy production, certainly, and actually I'll be touching on this a little bit more than I usually do. I haven't actually dedicated any sort of specific episode to this, but maybe I will one day. But I'll be discussing a particular different way to look at energy production in this podcast, or in this video. So stay tuned for that. And then I'm not going to touch on this too much, but it's also implicated in iron sulfur cluster formation, which essentially means that it creates these molecules that are based on iron. And those iron molecules are then used inside the mitochondrion as well as in enzymatic reactions. So proteins that allow for things to happen within your cells, some of them require iron. And that is where the source of iron comes from the mitochondrion. So that's a really critical role of the mitochondrion. Another one is calcium regulation. There's these different proteins, these different transporters, or what are called uniporters, inside the mitochondrion that allow for the exchange of calcium to regulate calcium within the mitochondria. And this actually has signaling consequences in that with particular signaling, you can get the mitochondrion to behave certain ways. I'm not going to focus too much on this, but it is touched on in the review. And if you'd like to read the review for yourself, you can. It's linked for you, so you can check it out for yourself. As well as my notes are also going to be listed there, which are quite extensive. And another one is apoptosis. Not that these four are the only ones, of course, but apoptosis is another one known as cell death. So if you have too many mitochondria that end up undergoing what's known as an apoptosis mechanism, where they end up releasing a particular molecule known as cytochrome C, then that accumulates, and then that leads to the death of the mitochondrion and therefore the death of the cell. So just to keep that in mind, I suppose real quick, I'll also add for the energy production, there are a number of different systems that are implicated in energy production. If I look at my notes real quick, just to mention that there's the TCA cycle, which happens within the matrix, the very inner section of the mitochondria. And this is going to be of some importance throughout this presentation. And you also have beta oxidation, which is the cleavage of fat molecules so that they can undergo the TCA cycle. And then there's the electron transport chain, which I'll get to in just a little bit. So therefore, it's pretty important. Mitochondria are incredibly important, and mitochondrial dysregulation, or the lack of function of mitochondria, is considered to be a hallmark in certain disease states. Now, I always hesitate. I mean, I do research in mitochondria, but to be honest with you, we've had ages of like, oh, this is the hallmark of disease, or this is the hallmark of disease. And it gets to a point where you just end up realizing that the cell is just, it's not really one organelle. It's not just one section of the cell. Usually you'd have to pay attention to many sections of the cell to really understand how cells end up undergoing disease. But for now, everybody's focused on mitochondria. So everybody keeps saying this is the hallmark of disease. And I'm sure it plays a huge role. I'm not saying that. But I'm always cautious because researchers like to inflate their own research as if it's the most important of all. And these reviewers may be doing that in saying that we're covering mitochondria, therefore it is so important and you have to know about this. Well, I agree that you should know about it, but if it's the reason for all disease, or for a majority of diseases, or the reason for a single disease, I'm skeptical of that if it's just by itself. But I digress. So one of the actions that mitochondria can undergo is fusion. So fusion is when you have actually two mitochondria that then end up tethering together and end up leading to the formation of a longer mitochondrion. And we've got that imaged here. Now here, this is kind of the cellular workings. You've got these two mitochondria as they're starting to fuse together, and then they end up leading to a longer, larger mitochondria. And this is going to be important because this is actually one of the hallmarks of metabolic regulation, as I'll explain in just a second. But before I do that, I'd like to introduce you to two different players when it comes to the molecular mechanisms of mitochondria. And so here, I took this from another paper. So the mitochondria here is in green, but they're not usually green. They're not even necessarily brown, but we'll just go with it for the time being. But these are synonymous, let's just say. Now, on the surface of the mitochondria, and on this outer surface of the mitochondria, in the outer membrane, there are these proteins known as the MFN proteins. These MFN proteins will then interact with the MFN proteins of the other mitochondria and kind of entangle themselves together. And that's through molecular workings. They end up kind of touching with one another, kind of holding hands almost. Now, once that happens, the two outer membranes will fuse together, and then you still have this inner membrane that's still part of the individual mitochondrion. Now, on that inner membrane, which is here, you have these OPA proteins. So these OPA proteins then end up entangling or connecting with one another, kind of doing a handshake together, and pull each other in, which leads to the fusion of the inner membrane. And then you have a longer, larger mitochondrion as a result. And that's exactly what we find with fusion, and it's based off of, largely, we look at these two types of proteins. They're two different MFN proteins, but we're just going to look at them in uniform as MFN proteins and OPA. So MFN being on the outer membrane and OPA being on the inner membrane. All right, here we've got our first kind of signal of metabolic regulation. And I realize this may look a little complex. You don't have to worry about really any of this stuff because we're not discussing the electron transport chain in any great degree at the moment. But what I did want to get across here is that, I'm looking at my notes for a second, that you have greater fusion, so greater amounts of this occurring, or this occurring, when you have more fat present. And this is the same fat that you would consume from your foods, these fat molecules specifically. Here I've got illustrated saturated fats, but it may also be the case with unsaturated fats, although the mechanisms by which these unsaturated fats enter the mitochondrion may be slightly different because they have to go through some enzymatic reactions that saturated fats don't have to. But that doesn't really matter. The point is that an increase in fat molecules around the mitochondrion or within the cell in general leads to greater fusion levels, so greater amounts of these larger mitochondrion. Now, why might that be the case? It's postulated that that's because the mitochondrion, or the cell in general, is trying to increase the amount of these proteins here, known as the electron transport chain. So briefly to describe this, usually you have an intact fat molecule that enters the cell, and then it goes through a process where it gets cleaved. This is all made of carbons. Little 2 carbons molecules are chopped off of this larger molecule. Then those have entered the mitochondrion, and they go through a process known as the TCA cycle. Now, off the TCA cycle, you get these products, these other molecules known as NADH and FADH, which are right here. That's why I have all of this displayed here. Again, not that you have to remember the electrons or coenzyme Q or complex 2 or succinate dehydrogenase or anything like that. But what I do want you to know is that from these fat molecules that are cleaved, you get NADH and FADH. Those then interact with complex 1 and complex 2 of this chain of proteins known as the electron transport chain. And those then go through a bunch of chemical reactions. That's not what I'm going to be going into. They go through a bunch of chemical reactions, and they ultimately lead to the production of ATP, or adenosine triphosphate, or really simply cellular energy. So the larger the mitochondrion, the association here is that the larger the mitochondrion, the more of these OXPHOS proteins, these electron transport proteins, which are also known as oxidative phosphorylation proteins. And therefore you get the greater production of ATP, or the potential to create more ATP. And because fats have to go through this system to generate energy, it makes a lot of sense. So this is a metabolic regulation of mitochondria. However, then one of the questions that people have asked is, what if we artificially, or if we genetically overexpress just these proteins without the fat combination? Do we still see then increases in the mitochondrion, in the size of the mitochondrion? It turns out that no, we don't. So this can be relatively certain that it's actually the presence of fat, the abundant presence of fat, that leads to larger mitochondria. Pretty interesting. I will say from my experience, there's a delicate balance here, however, between lipotoxicity and just having a little bit more fat around. If your diet is mostly in fat, for example, compared to if your diet is mostly in something like saturated fat. I'll actually be presenting, most likely, I'll be doing a video on lipotoxicity and its effect on mitochondria and the effect on the cells in general in a few weeks. So if you'd like to stick around for that, then subscribe to the channel. You know what I'm saying? So here we've got a metabolic regulation. Pretty cool, pretty cool. All right, so here I'm showing this same diagram that I showed earlier, the MFN proteins on the outer membrane, the OPA proteins on the inner membrane. Here we've got our outer membrane, here we've got our inner membrane. And in between these two membranes, as you can see, there's space, right? Between these two membranes, you have what's called as the intermembrane space. So just a space, that's all you need to know, that there's a space between the two membranes. Otherwise, there probably wouldn't be two membranes. And this MFN protein that allows for the kind of the coupling or the grabbing together of those MFN proteins from one mitochondrion to another mitochondrion, like we see here, for that fusion to start to occur, that is apparently sensitive to the cellular energy state of the cell, or the energy state of the cell. So that means if ATP levels change, if cellular energy levels change, then you get more fusion that occurs. And I'll be giving you a few examples of what that looks like, actually from a different perspective as well. Now, what's postulated here is that it could be that it's not actually ATP, but it's actually something known as GTP. So this is where I'm introducing a concept that I have not really mentioned on Physionic before, mainly because I don't want to confuse people. And GTP, although it is incredibly important, it's not seen as important as ATP. So mitochondria certainly do produce mass amounts of ATP. However, they also produce GTP. So they're not actually sure, at the point of this review at least, if MFN responds to ATP levels or for GTP levels. And OPA is the same thing, if it responds to ATP levels or GTP levels. And the leaning, however, by the researchers and by myself, is that actually responds to GTP levels. So ATP is adenosine triphosphate, so an adenosine molecule with 3 phosphates on it. GTP is a guanine triphosphate. So this guanine molecule is attached by 3 phosphates. And it turns out that MFN and OPA are GTP aces, meaning that they're enzymes, they're proteins that use GTP and end up breaking it apart or using it. So the odds are pretty high that it's based off of GTP levels. So if GTP levels rise or decrease, we're not entirely sure yet, that then has an effect on MFN, making it more active and therefore leading to this fusion of the mitochondria. Now, I added this here, this enzyme here, because it's incredibly important in the illustration of this ATP to GTP back and forth, in that this enzyme known as nucleoside diphosphate kinase will take ATP molecules and produce GTP. And then that allows for more GTP to be present. So you can assume that if you have higher ATP levels, you likely have higher GTP levels as well, assuming that this enzyme is plentiful enough. Now, there's another source of GTP actually that I mentioned before. Well, I didn't mention this specifically, but I mentioned the TCA cycle. And the TCA cycle is another area where you get GTP formation. So inside the mitochondrion, you have the series of enzymes known as the TCA cycle, also known as the Krebs cycle, and you get the production of GTP. So we've got multiple avenues by which mitochondria can produce GTP and affect both of these proteins. All right, so shifting our focus now to fission. We talked about fusion, which was the combination of two mitochondria into one lengthier, larger mitochondria. And now we're talking about fission, which is the taking of one mitochondrion and turning that into two mitochondria, smaller mitochondria, and that's fission. Now here we've got a series of different proteins that are actually involved in this process. The main one is DRP1. So dynamin-related protein is the main protein that we're focused on. And then there's a series of different receptors for DRP1. So these receptors, so we've got DRP1, which is kind of floating around in the cytosol of the cell, so around the mitochondrion, but not on the mitochondrion. And then you've got a bunch of receptors that are specifically on the outer membrane of the mitochondrion. And if those receptors are activated and DRP is activated, then DRP will then move from floating around the cell, or not around the cell, around the mitochondrion, excuse me, and will then bind to the receptor. One of those receptors is called Fis1. Others are called MID49, MID51, and MFF. Those are the big ones. Those are the ones that we know of, and those may be the only ones that exist. But the point is that there are multiple receptors for DRP. And before, I mentioned that there's the MFN proteins for the outside, and there's the OPA proteins for the inside. So once DRP then connects with these receptors, whichever one, if it's Fis1, MFF, MID49, 51, then it starts to aggregate, and it starts to create a collar around the mitochondrion, and it starts to squeeze inwards until eventually it pinches off into two different mitochondria. And then you get the system that sets up again. DRP leaves again, and you have two different mitochondria. There's a number of reasons why the cell would do this. We're going to be discussing some of them, but we're not going to discuss all of them. So this is kind of, I'll admit, this is a bit of unnecessary information, but I wanted to throw it in here because there's a part of this that's actually important, but the real specifics are not necessary. So here we've got our DRP molecules that are aggregated on the mitochondrion, or squeezing in, creating that collar. And here I've kind of zoomed into one of these DRP molecules, these proteins. And what can happen is that that DRP are regulated. So there's a bunch of different molecular regulations to DRP. The main one that people focus on is phosphorylation, so the addition of a phosphate to the DRP molecule. So you have a phosphate that can be added to a serine, so which is an amino acid. So this is a protein, therefore it is made up of amino acids, just like what you consume in your foods. And if you add a phosphate molecule to a serine at the location 637, then you get an inhibition of DRP. If you add a phosphate to the serine at location 616, you get an activation of DRP. So the activation of DRP would lead to this collar formation. The inhibition would lead to DRP just floating around the mitochondrion and not going to the mitochondrion to create this collar. But these aren't the only regulations of DRP, actually. There are other mechanisms like nitrosylation, where you add a nitric oxide, sumoylation, where you add ubiquitin molecules. You don't need to know these molecules. I'm just throwing them in there because for the people that want to nerd out and have a little bit of molecular biology background, as well as acetylation. But I'm going to be focused on the phosphorylation. And just the fact that there's phosphorylation, that's really the big takeaway that I want you to get from this, because we're going to be discussing it right now. So here's a pretty interesting mechanism here. We're talking again about some metabolic regulation. And again, I realize that this looks a little complex. This is what's called a G protein coupled reaction. So this is the outside of the cell here. This is the inside of the cell. not the mitochondria. And just to be clear, this is outside of the cell, and this is inside of the cell. And normally what you have for a G protein coupled reaction, there's other different types of reactions, in that the point is that there's a molecule outside of the cell that ends up attaching to the cell on the cell surface, on the cell membrane here. And then that leads to a series of changes inside the cell that leads the cell to then react a certain way. Okay? Kind of like, you can almost think of it like putting a key into the appropriate lock and then turning it, and then the reaction is that the door opens. So here we've got a stimulatory ligand, which is just a stimulatory molecule, a molecule that will bind here, and it will stimulate that things will happen inside of the cell. That's all you got to know. Here I've got two examples that I've got listed. So one of them is Forskolin, which I actually have multiple videos on Forskolin if you're interested in that, where I discuss, I think, fat loss, testosterone, and maybe one other thing. So and I actually discuss this mechanism in more depth in those videos, so be sure to check those out. So Forskolin is a molecule that can bind to what's known as this G protein coupled receptor, and then you have a series of, it changes the molecular shape of this receptor, and then that leads to an activation of this downstream protein known as adenylyl cyclase, AC. Now, adenylyl cyclase will take ATP molecules, which is the same ATP that we discussed earlier, the cellular energy, and will then convert those to cyclic AMP molecules. That's really, you don't need to know the names, just know that it changes it to something else. From ATP, adenosine triphosphate, to cyclic AMP. So now it's something else. So now as the concentration of cyclic AMP increases, it will bind to a protein known as protein kinase A. And through protein kinase A, once it's bound, it becomes active, which will lead to a series of other cellular responses, which will eventually lead to the phosphorylation of DRP, which is why we're going full circle back to this. Now, with the phosphorylation of DRP, the reason why I've got it in red is because we're actually looking at this one, the phosphorylation at serine 637, which was inhibitory, meaning that, and DRP is a fission protein, is the key fission protein. Therefore, if you inhibit fission, you are leaning more, pushing the mitochondrion more towards staying longer, staying larger. Now, there are a number of different reasons why they think that might be the case. They think that, for example, with exercise, you're lowering the ATP concentrations. You may be lowering in certain areas of the cell the ATP concentrations, which then leads to the cell needing to produce more ATP, and therefore it ends up producing more of these mitochondria. It's hard to reconcile that theory, though, because here you do need high levels of ATP. This may be based off of where the ATP is located, but you know, I'm just reporting what's in the review. I still have to put a little more thought into if I actually believe these explanations. But the point is that overall, you have some sort of stimulation that inhibits DRP1, and you get the elongation of mitochondria. This can also preserve the mitochondria from autophagy, or being degraded or destroyed. So this happens from the addition of Forskolin, so the consumption of Forskolin, but also from exercise itself. So these are two different ways that you have metabolic regulation, sort of metabolic regulation. This is more like cell signaling. I would say exercise is more of metabolic regulation of DRP1 and therefore the mitochondrion. Let me check my notes, make sure that I didn't miss anything here. Okay, yes. So now there are exceptions too that are based on the cell type. So what type of cells we're looking at. So we might be looking at muscle cells, for example. That could be the case. Here we're looking at brown fat cells. And I have some content on brown fat cells. I have other content that's going to be releasing in a little while. I'm not exactly sure when. I've got a bunch of other stuff I need to release first, and it takes a while to write the scripts and to think about, you know, how I want to write things out, and then to actually end up editing all this stuff while I'm still continuing my research and whatnot. So bear with me, but there probably will be more content on brown fat cells, because I know it's of high interest to people, considering that it leads to greater fat burning. Speaking to that, there are, I guess you could call them like exceptions to the rule, and this might be one of those exceptions. So in brown fat cells, not to be confused with white fat cells, the mitochondrion will undergo more fission. And the reason for that is actually sort of antithetic to what we were talking about with the increase in fusion to increase oxidative phosphorylation proteins, to increase fat burning. Because technically, the brown fat cells do want to increase fat burning, but they don't want to do it in the same way as what we described here. So here we've got an increase in fat consumption, which lead to the elongation of mitochondria. And here, we're not necessarily interested because of the increase in fat consumption. This is, I believe, with cold exposure, if I'm not mistaken. Yes, with cold exposure, you get an increase in DRP1 fission, or DRP1 and fission. And from there, then you get more of these smaller mitochondria. Now, why would you want that if you're trying to burn more fat? Well, you're not trying to burn more fat to generate more energy like you were before. What you're now trying to do is burn more fat to produce heat. So what ends up happening is you get more of these smaller mitochondria. This is all, again, kind of ideas, postulates that researchers are putting out there. You get more of these smaller mitochondria, which makes the mitochondria less efficient. And this is what you want in brown fat cells. You want less efficient brown fat cells, because it ends up shuttling fat into these mitochondria, and instead of generating more energy, you're actually wasting a lot of that fat. So you're breaking down fat, and you're getting rid of the fat molecules. So you are technically, in a body-wide sense, you are losing body fat, but you're not losing it in the sense that you're producing more cellular energy. You're actually losing it in more heat production, which is what happens here. This somehow, and I'm not clear on the mechanism, beyond the conventional mechanism of increases in what's known as uncoupling protein. But they don't actually mention that. They just say that it increases the inefficiency of these mitochondria, and therefore you see increases in this heat production. So in this way, you get more metabolic, or more regulation of mitochondria in their behavior. So now let's look a little bit at the proteins. So two of the proteins that I mentioned, two of the receptors that I mentioned that may be metabolically regulated to act with DRP in fission. So again, if you go from one mitochondria into splitting it into two mitochondria. And in one situation, you have the introduction of a protein known as AMPK. So AMP activate kinase is a protein that is actually heavily activated when you are in a low energy state. So if the cell is in a low energy state, then you get more of this activation of AMPK. As such, AMPK is also, it has an effect on all kinds of different things in the cell. I mean, it's a master protein. And from there, it also has an effect on one of the receptors that ends up interacting with DRP1. So it will phosphorylate, add a phosphate, to the MFF receptor, the protein called MFF. And then once that's phosphorylated, then DRP is attracted to this MFF protein, and then from there will undergo fission, as we see once I remove my image, as we see here. Okay, so there's another mechanism, however, through mid-51. Mid-51 has a binding pocket, so the protein has a particular shape that allows for ADP, so the adenosine diphosphate, the ADP molecule to bind to that mid-51 protein, and then from there it changes the shape of the mid-51 protein, and as it is a receptor, DRP will then bind to the receptor and then lead to fission yet again. So you have a number of different mechanisms through energetics, through metabolism. So ADP, I suppose I should add that, I forgot to explain this. ADP is the lower energy state of ATP. So ATP gets cut or cleaved, hydrolyzed is the technical term, to form ADP. So in a lower energy state, this is another low energy state, and this is another low energy state, you get the production of, you get the activation of these receptors, mitochondrial receptors, therefore attracting DRP for greater fission. So again, some of this is conflicting information, if you've noticed, because sometimes cellular energy state tends to increase fusion. Lower cellular energy state increases fusion, and in other instances it increases fission. So I think that there's a lot more information that needs to be figured out, but I think that the trend that I can say is that with fat consumption, assuming that it's not lipotoxic, meaning that there's not too much fat, or that there's that you're consuming too many of these unnecessarily negative fats, which is still, the jury's out on that, at least for me, until I dissect more research. That leads to greater size of the mitochondria, and this may be because they're trying to increase fat oxidation or fat breakdown, fat metabolism. Okay, so to touch a little bit on mitochondrial movement, I'm not going to get hung up on this too much, but this is another aspect of mitochondrial activity or mitochondrial behavior that is pretty interesting. And I'll actually have a separate video on how this affects fertility in men specifically. But for the time being, here we've got a neuron, which you might find in your brain. This is a motor neuron. You might find it in your peripheral system. The ability to move is all dictated by your neurons. And at the very ends of these neurons, known as the axon terminals, which is right here. And here we've got a dendrite, but we're focused here that this is a dendrite of another neuron. I don't have that imaged. But here we've got our axon terminal, which is the very end of this long projection known as the axon of the neuron. And in the axon terminal, you have a lot of things happening. You have the exchange of calcium, you have the exchange of neurotransmitters like acetylcholine. You have a series of different things that are happening. So you have a lot of energy use in this. So the need for energy is increased substantially in this area. Now, up the axon, you do still need quite a bit of energy, but it's not as much as in the axon, presumably. So as a result, the mitochondrion will, which is really cool, and I'll show you an image of this. I'll try to show you a video of this as well. The mitochondrion will actually migrate, will actually move from up the cell, so maybe up here or up here in the soma and the cell body, and move down into the axon terminal so that you have more mitochondrion present here. The reason for that, as you can likely imagine, is because ATP production needs to be really high. Since the energy need is really high, you need a lot of mitochondrion packed in here to produce a lot of ATP. So another way that metabolism, or the need for energy, changes this. So how does that happen? Well, it happens through these proteins known as kinesin and dynin. So again, I've got this is a rudimentary image, but if you were to zoom into this section here of this mitochondria and moving down, these aren't mitochondria, and they're just vesicles, but it moves through the same process. So these, let's say, kinesin proteins will attach to a mitochondrion and will literally walk it down the cell. And the video of this is really cool. Again, I'll try to show it to you in just a second. And they're just moving across what are known as microtubules, which you can think of as like kind of highways as they move down the cell. So let's see if I can get you this video. Okay, good. It did show up. All right, so here we are. We've got our image of, let's say, kinesin, and then we've got our vesicle, which in this situation, we're talking about mitochondria. And it just moves on down the microtubule right here. This is the motor protein, the kinesin itself. So pretty nifty, isn't it? Pretty cool. And that's actually based off of polarity, if anybody's interested, how that process ends up working out. So the mitochondria are attached, and then they move down here, and then they unattach, and then they float around in the axon terminal based off of energy need. Pretty darn cool. All right, so for anybody that wants a quick recap of literally not everything, if you just skip to this section, I always see in my analytics, I always see a spike here that people just want to skip to the overall thing, which is fine, but you're definitely missing out a lot of information. So a few of the highlights is that mitochondria split apart through fission and merge together through fusion. So we know that for sure. And fusion is present when energy is needed. This is a little bit debated still. I'm not completely convinced by this, but at least with the addition of fat, we saw that there was an increase in more fusion and a decrease in fission. And fission, although I didn't actually touch on this, and I might do this in the next video for next week, is fission is present in eliminating unhealthy mitochondria. So yes, this means that I'll be covering autophagy and how autophagy specifically targets unhealthy proteins within your cells. So if you're interested in that, let me know, because I'm really thinking about creating the next video, this next lengthy video on this process, how your cells are able to figure out which mitochondria or which parts of the cell are unhealthy and target those specifically while leaving the healthy things. Really freaking cool. Really freaking cool. And fission is present in eliminating unhealthy mitochondria as well as to reduce the fat metabolism efficiency of mitochondria in certain cells. We talked about how this was brown cells. And these things, while you do typically see patterns, certain cells are going to behave a little bit differently than other cells in how they work in their molecular mechanisms. So this is in brown fat cells. This is to increase heat production, which then leads to greater fat loss. And mitochondria will also migrate to sites of low energy, as we showed with the kinesin and dynein proteins. Okay, folks, that's what I've got for you. Hopefully you found this informative, and if you did, share it. You know, it's always cool to share science, especially with small YouTubers, small people that are trying to get out the correct information in as unbiased as a way they can. All right, with that, thank you for stopping by if you watched the whole 40 minutes of this, and I will catch you in the next one.